Awesome.
Hi, Adesh.
Thanks for joining me today.
Thanks for inviting me.
No, of course, man. I was ever since I talked to you about that random project that I wanted to do. I was super interested in the kind of work that you're doing. Okay, we're gonna take this from the just pretend that I'm like a smart friend at like a dinner party. Okay.
Yeah.
Okay.
So I should have brought some food.
No. I mean, I'm just gonna ask like just because I want to like sort of understand, sort of like how you got here to work, the kind of work you're doing now, and just like some way to build up a little bit.
Yeah.
But because when I was trying to prepare for this, looking at your papers and looking at your work,
Okay.
there's you've been involved in so many different fields, I would say, like space stuff, like something to do with like some with space.
Yeah. Okay. We can come back to that later if you want.
Yeah, and then age, and then like immunology, immune aging, which is very super interesting. I want to talk to you about that.
Okay.
And just like because you seem to have because of your you bring the methodology to most of these papers, right? If I understand correctly.
Yeah. So it's like a transitional type of approach.
Okay.
Yes.
Okay. Right. So then these authors who are working in all sorts of different labs, they want sort of the statistical and machine learning, or just statistical machinery, like what?
So the spatial stuff that we did, or that I did, I worked in that group for a year and a half.
Okay.
As a staff scientist involved or responsible for the data analysis.
Okay.
And they had a long ongoing research line with space research, with microbiology applied for space.
Yeah.
And then they come with data to me. And that's also the type of data that I used to work on. Yeah. So it was microbiomics data. So I generally work on this high-dimensional omics type of stuff. So that's how I started to do these analyses, because I worked there, but I had also
How did you get into the high-dimensional multi-omics?
Okay, so then we need to go really to the time that the animals were still talking, almost. I did a master in biochemistry, but I hated the lab work. Oh, it's a story that you will hear from more people, yes. And so I decided, okay, in my last year, I got introduced into bioinformatics, but the computational part, like the programming part, not necessarily the statistics behind it. I decided then, okay, after my master in biochemistry, let's do bioinformatics. So I did a post-academic in bioinformatics, and there I got, for the very first time, introduced to omics. And that was actually the age of microarrays still, so that's why I said long time ago. And with this, I started to get interested in it. I went for work, and I came back to it later during my PhD.
Did you always have more of a mathematics abstract interest?
I always...
I'm trying to understand why the lab...
Why the switch?
Why the lab work didn't
It's very tedious, boring work.
I can imagine it's very repetitive.
So it's very regulated. You need to be very strict with everything that you do. And at that time, I didn't have the skill nor the mental capacity to do that.
Right.
But I never studied biochemistry because I wanted to work in the lab. I studied biochemistry because I wanted to figure out how things work and how they work at the molecular or at the chemical compound level, let's say it like this. But I wanted to keep on doing something, and during that lab work that I was doing, I actually realized that there is a big component in data analysis that we miss. And that was even before the time that people were starting to do generally omics type of approaches, these large high-dimensional scales.
Was this during your master's, or what
was during my master's.
Okay.
So then I said, after my master's, I want to focus on actually the analysis of these type of things. Went for a while to work, went back to academia for a PhD, and there the topic was really like, okay, we have mass spectrometry data, so proteomics type of approach, quantitative proteomics. What can we do with it? How can we properly analyze this type of data?
Did you get the necessary mathematical training during a master or bachelor, or did you get it after?
Not in the biochemistry, but I did also one in statistics, and there I had all the skills and topics that I needed I could use. Okay. I had, of course, a basic mathematics training, but it's not too advanced. It's like just enough to do the statistics part, but also to do the methodological development. Yeah, so if you can do an integral, that's fine. A derivative is also fine. But I didn't spend ages on, or like 4 years on pure mathematics, no.
That's amazing. Yeah.
I come really more from the applied side, and that's what you also see reflecting in my research.
Yeah, it's interesting because I think people might have the idea that if you wanted to do a sort of a fairly abstract and mathematical the data science contribution to this to the biological domain, you have to either have be extremely focused on the biology or have this supremely deep mathematical background. Do you think that's fair? Or
I think from my experience, from the people that I worked with people who had done the mix like I did, people who had a pure biology, people who had pure mathematics. What I do see is that for mathematicians, it's very difficult to talk to biologists because they don't understand all the ins and outs.
Okay.
For a biologist, the opposite, yes. I mean, they understand all the ins and outs, but they have no clue how to come to the right derivation of a certain model or analysis type of thing. I do feel if you have a decent background in mathematics, it's easier to make the step from biology towards statistics, let's say it like this, than from a very heavy mathematics-oriented background to come to real-life solutions, yes.
Okay.
There is not, it's possible, yes, but it's very difficult because you need to learn to speak a different language.
Right.
You can come up with a great solution, but maybe it doesn't solve the issue that the biologists have.
Right.
That is, for me, my advantage, that I do get what the issues are for the biologists. And what I make up with that, I can actually spend time on developing the mathematical things then to the level that I need to fix the issue.
Yeah, so this is quite similar to the sort of the path that I took to end up here, because when I did my very first college education was as an electrical engineer.
Okay.
So I finished that. Then I went to work, and then I spent 5 years just doing random jobs, and then I just missed the technical sort of research. Like I just missed it. So I went back to school to study AI, and then 5 years of AI that I studied was very broad, a lot of mathematics, no real biology, just mostly cognitive modeling and language modeling and statistics and machine learning.
Just really the background and why do these things work.
But luckily for me, the second year of my master was super flexible, so that I could basically make it however I wanted it to be, however I wanted it to be. And basically in the first year, I decided I want to go all the way, like do biology type of work. Then I got an internship at technically TU Eindhoven, working on computational biology problems, and then did my master in EEG processing and predicting mental health things and that, and that's how I sort of like I built the ramp onto the biodomain, and then I sort of learned the biology on the way.
It's possible, yes, but it's not easy, yes.
Because
you come from a technical background and a mathematical background, and but that's what you need, yes. You need to have understanding of the methods, but also understanding of the application area.
Yeah, exactly. So it's funny because for my thesis, I used the LLMs a lot during my to when I was writing the code to build things, right? Because I said differently. I really wanted to use LLMs. I really wanted to use the best LLMs to make it faster.
Okay.
But they were so not trained on, for example, this super niche transformations, super niche representations of EEG signals and these things, and they would try to apply generic mathematical transformations to the data sets, which makes no sense from the context.
But is it also not because they're not necessarily trained on that specific data?
Exactly. Yeah. So the more I got into my thesis, the less useful they became, because they just kept kept proposing ideas that just didn't make sense given the context of the data. Because for them, it's just a matrix. So like a matrix comes in, these functions should matrix out, but you can't just arbitrarily do a transpose just because to get the math to work,
Yeah, I understand, yes.
So that's very cool. Okay.
So there are things in common, yes?
Yeah, definitely.
That's nice to hear.
That's really good. Okay, so you started off with mass spec data.
Yes.
First, what is mass spec data?
Okay.
I know it's a measure. I know it's a way to measure the weight of it.
To simplify it maybe to the extreme, it's a very expensive balance.
Okay.
With the extra addition that it not only tells you what something weighs, but also how much is present.
Yes. So x-axis you have
the mass,
and the y-axis you have the abundance on
yes.
how much of a particular mass is existent in your sample, or
so like for instance, if you do proteomics, so you take proteins out of the tissue that you're interested in, you put it on a column. From the column, because you want some separation, It's easier for the mass spec to handle it then. You put it through the mass spec and you get on your on your z-axis, your x-axis, here we are, third one, you get an m over z, so the mass over the charge.
Okay.
Because you ionize the protein or whatever you put, the protein or the peptide, what you put in, so it can get multiple charges. So you get a mass over charge value. And then the y-axis is telling actually how often the intensity of the signal, how often it detects that specific ion.
I see. Okay. And then so if you have a protein, if you want to get an idea of how much of a protein is in a sample, or
the difficulty with mass spec or with proteomics or anything what you load on a mass spec is that you do not necessarily know what you're measuring.
Okay.
Like with genomics or microarray transcriptomics, sequencing transcriptomics, you map your reads back and you know this is this gene, and you do your. So with the mass spectrometry based proteomics, you do need to do something similar based upon the masses that you see. You need to try to identify what protein it is or which peptide it is, and then you get, in the end, you see for that protein, you see this intensity, and you get back your data matrix or your data frame.
So you have a prior map from mass to protein,
It is a bit more complex. What you do is you have, it's called MS1, MS2. So in MS1, you have basically the protein that you measure or the peptide you measure. In MS2, you take out that protein and you break it into smaller pieces. You do the fragmentation. So you get an MS2 spectrum, which tells you more or less which amino acids are present and in which order.
Okay.
And this you map back to a database, or you do it de novo, and then you say, okay, this is my protein. These peaks correspond to that protein, so I now integrate over the extracted over the ion chromatogram.
But don't you lose in this process? Do you lose the three-dimensional shape of the protein when you just break it down?
But you're not interested in the three-dimensional. You're not interested in the structure. You're interested in qualitative presence or in quantitative presence.
Okay, so
basically.
the data you would use for a 3D problem is different.
You can do with mass spec also structural proteomics, structural biology type of things, which I'm actually quite interested in myself. But most of the time, it's just used for what is present and how and to what extent in quantitative approach.
Okay. And I can imagine how valuable it is to be honest, I didn't realize how difficult of a problem it is to actually measure biology at that scale. I just thought you just sort of look at it.
Okay. That's one way. Now a mass spec actually in its core is like an incredibly fascinating technology. How it works, it's super complex. You have really a combination of pure theoretical physics coming into play, chemistry, how do these things break up, also biology. So this is why you see in mass spec field very often people coming from different backgrounds actually coming together to do proper mass spec.
So is it just like a machine that you just pump this thing through, or is it like multiple people have to sit there and do a bunch of stuff?
It's one big machine, and then you have in most labs which have experience with mass spec, you have two people working on it. So you have one person who actually tries to gather the biological information, the biological data, so the sample, extract the proteins, and that's given very often to the technician who's actually running the machine.
Okay.
Now, if you do a PhD, you will learn both of these things because, those labs are not necessarily so big. But if you go to pharma, for example, or you go to these analytical labs, also for pollution type of things that they use them even at the airport, actually.
Oh, you mean to detect the substances when they
so they swipe you, and they put this in that machine. That machine is like a mass spec.
Oh, okay.
But that's like probably one with gas chromatography. But the ones that I'm used to work with are the mass spec is coupled to a liquid chromatograph. So you do the extra separation. Instead of doing over gas, you don't do it in gas, you do it in liquid.
Oh, wow. So they have turned the technology into a super simple interface that you can put it in an airport, and someone without any special training can just do a mass spec.
Yeah, they swap you and they just know I need to put it in this port and then push around and the thing will do it for you. But these are, dumbed down is maybe not the right word, but they are very simplified approaches.
Yeah, so I mean, it's probably like a piece of technology that gets you 80% of the way with 20% of all the effort. Not good enough for high level research grade research.
No, but that's also not needed.
Yes,
if you detect something special, then you can go further on.
Right.
But they send these things also to space, for example. Yes.
Right. So they would the machine just basically checks for the abundance of some of the
Yeah, so
chemicals it cares about.
So what basically happens in the background is you ionize a protein, yes, that comes off the column, or a peptide, which is a subpart of the protein. You ionize it, yes, and then you actually put it through something that actually will select something with a specific M over Z value.
Right.
And then you take a long time to measure it, and then you get an extra separation. So it will do it's like an extra level of separation that happens over mass, sometimes over shape. Then you add something extra to the mass spec. And in the end, you have a detector that detects how many ions actually ended up at a specific time point that corresponds to this mass to the detector, and that's what your y-axis will be.
So this is at a molecular level.
That's purely at the molecular, if it's present or not, basically, but you see it over a time-based type of thing, and then you see how often it's
Okay, so, and there's a unique mass number for every molecule? Or
every protein or every peptide consists out of these amino acids.
Yes.
The combination of these give you relatively unique masses.
Interesting.
But because not every protein has a unique mass. They can have overlaps, for example, especially when you cut them in peptides, you can have overlaps. So that's why you will do the fragmentation. You will break the peptide or you break the protein into smaller pieces, and you see which amino acids come off, and then you get an ID. It's probably protein A, or it's the isoform of this protein, for example, that is present.
Okay, nice.
So it's like a very expensive separation balance type of thing together.
So it's like, if you think about it, it's something like you do with fractionation of blood, where you take the blood and you break it apart in different places?
You take the blood, you try to pull out all the proteins that you need from the blood, you put that on your liquid chromatograph, or if you do gas, you will put it on a sample and you will heat it. And that is being pushed through. So you already need to have what you want to measure up front, you already have collected. Yes, that's what you have a technician for. And then there is a technician who runs the machine.
Okay, nice. All right, so you started with this... This is your introduction to high dimensional data stuff.
That was during my training. I got introduced to mass spec already. I knew it, and then my project for a PhD was specifically focused on specific labeling technique to do quantitative proteomics. But as always, a PhD digresses, and you do different other things. Yes, but this was the very, very first, for me, hands-on research thing, like data that I was working on.
Brain. Did you do that?
Yeah. So
during your PhD or
no
PhD? Yes.
Yes. So the very first project I got was structural biology, mass spectrometry in combination with structural biology, and we got stuck in a computational issue.
Okay.
But my promoter, who was half-time or like partially working in Poland, partially working in Hasselt, where I did my PhD in Belgium.
Yeah,
then
put this thing down a little bit, the mouse.
Yeah, sorry.
No, it's good.
Is it better?
Yeah, better. Yep.
Okay. So he was in contact with other people. In Poland, for example, he was teaching their statistics, and he got to know a group where we were also interested in mass spec. And there was a PhD student who did not have any topic. The way that it works in Poland was apparently different than I was used to. So he came over for a research visit. We introduced him the problem. And he came a few weeks later. We had a very good background in computational sciences, very good background in mathematics. And he came with what he thought would be a solution for a computational problem. But when he was explaining it, we realized this is not the solution that we need, but this is actually something related to how you can calculate the signal that comes out of a mass spec. So you can calculate an isotope distribution. You like carbon, you have two isotopes.
Yes. So an isotope is the same. It's like a small variation on the same element, or Okay.
So I need to be careful now because I can mix it up. It's the same atom, but it has a different neutron content.
Okay.
So if carbon 12 and carbon 13,
okay,
the difference is 1 neutron.
Okay, and that
has a mass shift.
Mass shift. Okay.
So if you look at the signal nowadays, especially if you look at the signal from a protein, you will not see one peak, but you will see a series of peaks. And these series of peaks are specifically for the composition, the elemental composition of that protein. So no longer the amino acids, but how many carbons, how many hydrogens, and so on.
Okay, I want to ask a dumb question.
There are no dumb questions.
Can you look at a protein under a microscope visually with your eyes? Or is it too small?
It's too small, but people do cryo-EM and they are able. That's how you can actually see structure now.
Okay.
So you can do it in that way. So you need a very powerful... not telescope, that's for SPARS, but a very powerful microscope, yes.
Okay. Cool.
So he came up with a solution to do the calculation, and we said actually that not fully solves the issue we had. It was a complex combinatorial issue, but looking at it, and then my co-promoter who worked on mass spec as well in the past and was interested in isotope distributions, he realized together with the other people on the table, including me, this is a trick to actually calculate an isotope distribution in a fast, efficient way. So instead of having the solution for the problem that he came, we found actually something new. So we found a faster, more accurate approach to calculate now.
How did they do it
isotope
before?
distributions.
So before this method, how do you calculate the isotope distribution exactly?
Okay,
So first of all, why is it important to calculate the isotope
yeah,
distribution?
so if you can calculate the isotope distribution, you can say something about the shape of the peaks, and you can already help your identification.
Okay.
Yeah,
So identifying
you
what
can-
protein it is a huge problem.
Yes, like if you, I might be off now because it's been a few years ago that I looked, but the identification rate of proteins and proteomics with mass spec is 20 to 30%.
Okay. Well,
Meaning you lose a lot of data, yes, because of fragmentation doesn't go the way, or some things are not fragmented. I think they push it higher now with different techniques, like different way of fragmentation.
and if
so
you,
on.
and just one more level up, you use this. The reason you want to identify proteins is because for things when you're doing a blood test or doing some sort of identification of a cell type of a
You
cell.
want to use it to
cancer-
understand the biological process. You want to build biomarkers, the same thing as you would do with transcriptomics. Yes,
Okay.
which genes are active. So now you want to see which proteins are present. Okay, you need to have as much proteins as possible that you can measure. Instead of measuring 10,000 proteins, you measure 2,000, 3,000. Although you know there might be 15,000 present.
Right.
So you want to improve that. There has been done a lot of experimental work on that. The people start to report higher percentages.
Man, okay, wait. That's a huge. So if you let's say you're looking for a specific protein in a sample,
Yeah.
you could have 15,000, 20,000 different proteins floating around in that sample.
Yeah, what you can do if you want to really know one specific protein, you can of course try to purify it before you pass it to any
Okay.
analytical technique. Yes, but if you want to do really omics, like the study of the whole proteome or the study of the
Just to
whole,
see what's going on.
just to see what's going on, yeah, you get a huge amount of proteins. Yes,
Got it. Yeah.
I mean, think about we have about 15 to 20,000 genes depending how you count. We have tens or hundreds times more proteins.
Right. Because each gene encodes for a process that generates some kind of
Well,
protein.
each gene actually.
What is the
codes-
mapping there?
So yeah, it's not really clear, but each gene has different isoforms have different forms, and each isoform is corresponded to a different protein. So it's I think 1 to 10, 1 to 100 or something.
Yeah,
like
I'm still getting wrapping my head around like from the pathway from gene to protein.
Yeah.
Where you do the DNA, like the whole process.
Yeah, so that's the central dogma. Yeah,
Yes.
so you would have DNA for human beings. Yes,
Yes.
we're not going to talk about viruses or like some things that can do the opposite. You go from DNA, you actually transcribe it,
Yes,
so it
to
becomes
RNA.
messenger RNA.
Yeah.
There are steps in between, but that's ends up to the messenger RNA. This is being fed to ribosomes, which actually produce the protein for you. So it will start to read triplets of your base pairs, yes, and that will generate specific amino acid sequence, and this will start to fold and form your functional protein.
Yep.
Okay, so one gene can have multiple isoforms. So depending on where the introns and exons coding parts and non-coding parts of your genes are, you can get a different combination that will generate different isoforms. Additionally, if something goes wrong with that can also happen. So when you go from the messenger RNA up to the not to the DNA, but up to the amino acid sequence, something can go wrong.
Mm-hmm.
But what you also very often see is once you have the protein working in its multiple isoforms, you have like for one gene 5 isoforms, there are also post-translational modifications happening. Phosphorylation, for example, is one typically known. Acetylation is another one. This might activate or deactivate certain proteins or push it to do something slightly different than what we know.
And this is all before it. This is happening all inside of a cell that's generating this.
That's all happening in a cell. There are post-translational modifications happening outside the cell if that protein has an extracellular function, yes, outside of the cell function. So to really say what the overall number from one gene to the proteins is, it's huge.
Okay,
That's the only thing you can say. Certain ones are very conservative and very well conserved. One gene, one protein, but some of them give a huge range.
Okay, so some of the proteins that are generated are for the cell's own internal things to happen, and then some of them they put out of the cell into go do other things like hormones and things like this. Is that?
Yeah, indeed.
Okay.
And you can get different modifications on top of. You can get sugar getting bound in it and these type of things. So it's a super huge research area that you can look into. A super huge number of molecules that you actually start to measure.
Okay.
Mass spec helps you.
Okay.
But identification is lacking behind.
But do we have sort of a pretty good map in terms of all the proteins that exist?
No. That's
in
one
the
of
body? Like in the human body?
No. Well, we have an idea what the base form should be, yes, and also with the isoforms, because you can predict that if you have the DNA code, you can predict what the proteins would look like.
Right.
But there are so many other biological processes going on that actually lead to the phosphorylation or to the glycosylation, adding the sugar on top of it, that it's very difficult to say, we know from a human being we have 150,000 proteins. I don't know, it could be 1 million as well. There are people focusing on different type of things, but the base forms, I think we know to some extent, but it's all the modifications. And these are actually the ones that we are interested in because they are generally the ones who drive the biological processes.
Okay. Right. So, wow. Okay, sorry.
So there is an extra level of complication.
Exactly.
Transcriptomics in itself or genomics and epigenetics is already super complex, but with proteomics it becomes even more complex. And then if you want to go a step further, you look at metabolomics, so your protein does something, there are breakdown products, yes. Mm-hmm. These are even more complex. So there's an even larger variation that you would have there. And the alphabet also changes.
Yeah.
Like you have for DNA of ACGT, you have 21 amino acids for a human being. Okay, there are some extra special ones, but let's just keep it on 21. For metabolites, you don't have that anymore. There you have carbon, hydrogen, nitrogen, and all of these type of combinations. So the complexity increases the lower you go into from top to bottom, from commanding an action to actually the end of the action. You have multiple sublayers. The lower you go into this pyramid, the more combinations you have, the more complex things become.
Okay. Wow. Yeah, okay. Sorry, we got here from asking about your...
from my background up to what a mass spec is.
Yeah, your brain, the brain...
so the brain paper is a way to calculate if you put a protein or like even an organic compound or like a metabolite or something like this in a mass spec, how would the signal look like? And that signal we call the isotope distribution. So brain, the Piotr who developed this, he found a very nice way to quickly calculate it, because the issue is, the first peak we call the monoisotopic piece. That means it's consisting of elements that only contain the low isotope variants of your gene. So it's carbon 12, hydrogen 1, and so on. The second peak, this is the M+1, it's generally 1 Dalton further. That contains one isotope, but it can be the carbon isotope, can be the hydrogen isotope, can be the nitrogen. So you start to see, it becomes a complex computational problem, combinatorial problem. Yeah. So what he found was one very intuitive way actually to calculate this.
Okay.
By looking at number theory, basically.
That's amazing. So just a subtle difference in, I don't think is the difficulty with this whole this field of work is that it spans from physics at the very lowest level to all the way to biology, and then you have the mathematics that translates from the different levels. Mm-hmm. Man, it was super. I made no progress trying to figure out.
I know your feeling, because people who start to do something in mass spec, they come from different angles. It takes a long time. Yeah. If you go to the biggest mass spec conference, it's the American Society of Mass Spectrometry organizes every year, 8,000 to 9,000 people, and you see people coming from all different areas. So you have sessions on people who build a new machine, people who actually do really clinical trial clinical stuff, almost clinical trial stuff. You have people do the pre-processing, computational scientists. Everybody comes together there.
Wow, that's super interesting. Because that's very interesting. Because, for example, if I go to like a machine learning conference,
most of the time only machine learning.
It's very interesting, man. So when you want to contribute, you can pretty much pick. You can be a mathematician, you can be a physicist, and you just have to somehow work together and come up with the.
Of course. Yeah. So this is what you see, not only in mass spec, but you also start to see this in other omics. Yes, that also people coming from the wet lab themselves are developing methodologies to do the analysis.
Right.
Not always in the right way, because they don't necessarily have all the time the right training. But anyways, you see people with different backgrounds actually trying to do the same thing, and it's really multidisciplinary type of research that you see happening for the computational part or the analytical part. Let's call it like that.
Okay, all right. So you started with mass spec as your gateway drug to.
Yeah, that was my first real introduction in omics, to do it myself.
Okay.
I've seen how you would do microarray analysis, but I never really did it properly.
Okay.
So research-wise was mass spec. At that time, then I got a question, because we had to also do some consultancy.
Mm-hmm.
Came a question from a lab that I was not working for as a master student in biochemistry, but I did very related work. With the question, we have sequencing data, and we would like to do some type of the step before you do association studies. We would like to find out which potential genes are linked to something. Could you help with that? So I got that question, so I started to work in sequencing data too. And until now, this is the combination that I'm doing.
Right. Wow.
So it's or sequencing or mass spec stuff.
Okay. So what is like the cutting edge right now in terms of like in the mass spec sort of research?
Well, the difficulty with mass spec is not only all these issues that I talked about the biology, but also people come up with new machines every so many times. So you see every 2-3 years there is a new machine, meaning the output will be always the same, but it's higher resolution, more sensitive, less background noise. So the pre-processing always needs to be altered.
Okay.
So most of my research focuses partially on the research, partially on the pre-processing, and a tiny bit also on the, two different groups, do we see a different yes or no? Or selection of biomarkers.
Okay, nice. So, like for people who don't live in this world, like what does a biostatistician do at a medical center actually do day to day? Like what does that look like?
For me particularly or in general?
No, for you personally.
Okay. I start with a coffee. Okay. It's also important. I need to get the brain to activate. So what I generally do is I try to support people with their medical research in order to help them to make sense of the data that they generate. Because I'm focused on high-dimensional data, on omics type of stuff, I'm talking to molecular biologists or to people who study, for instance, metabolic diseases. So they generate these large data sets. Per person have thousands of observations, yes, like gene 1, gene 2, gene 3, or protein 1, protein, and so on. And I help them to make sense of that type of data, because it has become very complex data. It's not always so simple for a biologist or a doctor to interpret that data.
Okay, so how much of machine learning sort of predictive tasks are versus purely sort of statistical analysis and significance testing and these kind of things?
Yeah.
How much of it?
So there are like two directions. Yes, you can do the inference, so really seeing there is a difference between two groups, yes, and we see which are different and can we build a pathway from it or like build a network from it. The other way is the biomarker detection, and especially there the machine learning comes into play.
Okay, so the machine learning algorithm is the detector basically, or
the machine learner will actually, based on the data frame or the data matrix that you get, actually will tell you this feature is important for discrimination between these groups, this feature is important as well, this one is not so important, and so on.
Yeah, because when I was thinking of machine learning, I was thinking of, say, a deep learner, and then
but the deep learner can do that too, yes.
Yes, but I was thinking, one of the things that is interesting of many things of a deep learning, say just a standard multilayer perceptron, right? Just a deep learning thing, is that its job is to transform the input into any shape the shape that it needs to make the prediction better or worse, right?
Yep.
And I know there's a lot of work, for example, in CNNs that was done to actually see what those internal representations look like to make sense. And I was just thinking,
so this is like you're looking at the perceptron, for example.
Exactly.
Yes? Okay.
And if you think of, so if we have the feature, if we start with the features that we know, so we're putting in a matrix, right? And then we want to see, this feature, how much does it impact the prediction this much? But then we are sort of starting from the assumption that one of these things are important biomarkers probably for this to work. We just don't know what it is. But internally, it could be that there's some abstract combination of them that is actually the biomarker that we just don't actually know yet.
Yeah, so what you see in clinical practice, people do a blood test.
Okay.
They know, okay, if we see these and these certain things elevated, then means it's probably this type of disease. But it's already a convolution of many molecular agents that is happening, yes?
Mm-hmm.
Or like you go to the doctor and you say, I'm tired.
Mm-hmm.
You give a number of symptoms based upon those symptoms. But these symptoms are a result of all these molecular actors that you have. So if you look at the omics type of thing, you try to figure out what are these molecular actors. But because their interactions are so complex and you have so many different possibilities, it's very difficult to really understand on that level what is going on. And a deep learner can help you because a deep learner is actually trying to combine them, yes, find non-linear relationships, linear relationships, and so on.
Yeah.
But that's what the difficulty is actually about.
Yeah, it would be amazing. Before I came in, when I was first getting into sort of interpretability and sort of trying ways to get inside these different layers and see what they're doing, I wasn't fully convinced that why is this so important? Because I thought you got a good model, you can get good prediction. Like what is the real argument for this? But now I'm thinking, okay, well, once I started to sort of get a feel for the complexity underneath, that has to be sort of brought in together to make this thing work.
Yeah, you were looking in the past more on, okay, does it predict well or not, without actually specifically looking why do these features are part or in which shape they are part.
Yeah, that's
that's what molecular biologists are very interested in, because if they understand how the mechanism works, then they can actually try to figure out, okay, what is important and what might be potential targets
for the
to develop therapy for example, yes?
Right.
So in that sense, it starts to overlap again a bit with inference type of things, or like trying to make sense out of the data. But there are two different approaches to do that, yes.
So one thing, I'm just saying things that come to mind. So let's say a deep learning model learns some series of transformations to make the prediction better. And you say, okay, so we have these features, we have this thing in the middle, then we have this output. Somehow the features that we chose here are important to predict the output. And
yeah,
so
hopefully, yes.
Yeah, so let's just assume it's doing a good job, right? And it does a really good set of transformations.
Yeah.
Isn't that just one in the space? So there's no saying that what's actually happening. Let's say you have a collection of metrics you have from the body of or blood sample. There's no actual way of saying that the actual process that's going on inside of your body is anything like what the machine learning model learned just to make the prediction happen.
Yeah, no, it's a different optimization function.
Right.
If you really want to make it very theoretical, it's a different or abstract. What you try to do as a living being, yes, is try to generate enough energy to keep on going on, yes, and eventually reproduce, if you would say it from evolutionary point of view, yes. But what your machine learner does is trying to optimize a different function, a different minimization, different optimization, yes. But hopefully they coincide to some extent.
Right.
Yes?
Yeah,
or
of course. Like if you think about all the space of possible things that could happen, and the machine learning model is somehow able to make a prediction based on the features you give it, then it's probably not super far off in terms of what could be. Like all we know is, I think, I was a bit more bullish, a bit more naive about machine learning before, because I was from a purely machine learning
that's your background,
right?
yes.
Yeah. So we were all just like, number go up, let's make it happen, like get it to work.
Yeah.
And now I'm thinking, okay, well, actually, it says more to the fact that we have been able to say, look, these features are already important. We're going to pick them out and put them in the model. And then that helps the model, of course, do the recombination and make the abstraction and make it happen. But so much of that comes from, if you don't pick the right features, pick the right data, it doesn't matter how good your model is, it's not going to do anything, right?
Yeah, no. If you work for the hospital, they not necessarily care what type of model that you use. They just want to have something that is performing well, but is also interpretable, that they can link back, yes. Okay.
Even for this scale of problem, they still want to be able to say, hey, this is the real, okay, imagine you have a really super good model, and you have...
Yeah.
For example, if you can quantify uncertainty, and you can give some statistical guarantees, is that usually enough or not?
Yeah.
That's usually fine. So it doesn't matter how...
For clinical practice, it's enough. If they can say with 99% sensitivity and specificity, yes, that person has that specific type of disease, they're happy with that.
Okay.
It's the people who are going to do the research in the background.
Okay.
They would like to know, okay, why is this specific gene or why is this specific protein actually in there?
Right.
And that's what you try to do, yes, with the interpretable type of thing. For which subset of people, for example, this is more important than for the other. Yeah, think about the Shapley value story. That's there focused on, yes.
Yeah. I've been thinking a lot about when I've my first semester of education in machine learning and AI, the first thing you sort of hear is offhand, because I get this vibe that machine learning the community, they know how much of statistical knowledge they're using, but they have to do due diligence and like teach you some of that stuff. So at least one semester you would say, okay, well, linear regression, if we align, we try to, and they try to emphasize a little bit about why a simpler model is good, it's more generalizable, it's more blah, blah, blah. And past few days or so, I was really, but then I realized I was not really told actually why. Why is a simpler model so much better? And I was thinking about, okay, well, if you take you can tell me if I'm wrong, okay? This intuition.
No, go ahead.
An intuition that I wanted to see if I can get some input on, okay? So if you take a linear regression, let's just take a single output y, and you have one variable x. Just
the most simplest form. Yeah.
A univariate regression. What we are assuming is that y is the intercept, say beta naught plus the coefficient beta 1 times x plus some error.
Yeah, you have noise. Yes, what you cannot explain. Yes.
So I didn't really think about this equation that much, and then I thought about it. I was like, wait a minute. Okay. So on the left hand side, the y is a random variable, right?
Yeah.
That's the whole point. Like,
It comes from a distribution with some noise.
Exactly.
Yes. Yeah.
So we have a random variable. The beta naught plus beta 1 times x, that's just a fixed number.
Yeah.
And all the actual randomness comes from the error term, because you have to, it has to balance, right, the equation. And then I thought, okay, wait a second. So we have this, let's say we have some complex system. Okay, we get some set of one variable. And we're saying, okay, everything we don't understand about the world, all the noise, all the measurement error, all the randomness, all the complex interactions, all that shit, we don't care. Put it in the error term.
Yeah.
And then we say the relationship is linear. And then if you fit the model and you actually find the real relationship, and then I thought, wow, if you're making such a strong claim, right? You're saying all of that randomness, the variation, all the complex dynamics you think should matter for this super cool system, no, it doesn't matter, we put it there, and then we fit it with a linear line, and then it works. And you're just like, so that sort of like hit me. I was like, oh, I get it. If you can make it work by relegating all the complexity to an error term, does that make sense? Is the illusion make sense, or?
So if you would have the simple linear regression, you have the beta 0 plus beta 1 times x, yes, that will be the average, yes?
Yes.
And then the rest is put in the variation, yes? Now, where comes the problem with the overfitting?
Yes.
So if you would put many more x's in there, so you would beta 2, beta 3, and so on, you start to actually peel off of the variance of your error term, of your residual.
Yes.
At a certain moment, you will start to see that what you actually are peeling off is random noise.
Okay.
Or is not, it could be just a feature of that specific subset what you're looking at, and not necessarily is generalizable.
Right.
So that's why that's Occam's razor, yes, that's the whole theory about it, yes. So you would like to peel off as much information as you can,
right?
But it should be still quite okay. And the more information you start to peel off, the chances that actually what you're doing is perfectly working for that one data set.
Right.
But another data set in the same area might have slightly different features, and then actually might be slightly different properties.
Yeah.
And then adding more and more features on it, you will see you at a certain moment you reach like a maximum, and then you start to decrease again.
Right. And the reason why, for example, the linear regression is so difficult to overfit, is it because of the strong linear?
Well, simple linear, it only has one parameter. Yes, that's one variable. Yes. So it starts to overfitting comes an issue when you start to add hundreds of parameters.
And then you basically try to assign meaning to possibly random noise that's in the data?
Yeah, so let's say that you want to predict how tall somebody will be, yes? Now, we take age of that person into account, and we take the gender into account, yes? Okay, already with age, I'm making a mistake because it's not a linear function, but let's assume that it's a linear function, or we did some transformation that becomes linear, yes? So you can only take age into account. Yes, so you would have for female and males exactly the same thing. Yes.
Yes.
Okay. Then you add a second term, which would be gender. So this will be a categorical one. So you get two parallel lines.
Yes.
If you want to have the interaction, you can have non-parallel lines. Yes. And this, what you're doing is continuously peeling something off of the residual.
Okay, I see.
Because the two lines will give you smaller residuals, probably, than if you would use that one line.
Right.
Add a third eye color. I don't know, probably has nothing to do with length, yes. But that could be maybe in your data set still having a very small significant effect.
Right.
But overall, it doesn't have a significant effect. So there is where the overfitting starts to occur. Yes, you start to add variables that actually are explaining something that you not really need to predict what actually the outcome would be.
Right, because what you care about is not just this little sample, it's just the general population or the general problem that you're trying to predict for.
Indeed. So, we cannot measure the whole population, so you take a subset.
Yep.
By chance, they have slightly different properties than the whole population.
Yep.
Your overfitting will actually focus perfectly on that subset, will do perfectly, but it's no longer applicable for your full population.
Right.
For example, the eye color effect, yes.
Okay.
Or the lunch that you had in the morning,
and
for
this
example.
is why a neural network, which has millions of parameters, is more at risk of doing this overfitting.
Well, not necessarily, yes. Even just with the standard linear regression, like Tinker-Lasso model, yes.
Okay.
The classical, it's also machine learning, but the classical prediction, yes, and the statistical, they can also suffer from that, yes. That's why Lasso is there, why you put the penalization parameters.
So you send parameters, you shrink the parameters.
And that's why you do cross-validation, or why you have an independent validation set, yes, just to avoid the fact that you actually are over-reporting the accuracy of your model.
Right, and I guess that's where a bit of the art comes in, right? Because you have to sort of, it's definitely a bit of art, right? Like, how do you know how many parameters is enough? You just have to.
You do not know. Yes, you need to take a look on the outcome, but if you do it in the right way, that you take the risk of overfitting into account by doing cross-validation, nested cross-validation, multiple validation or test studies, yes,
right.
You hope to limit the effect of that you would have overfit.
So how do you deal with this when you have many dimensions of data?
You just put them in the model and you see which ones come out. Yes, if you do Lasso, there is an upper limit, and that's depending on the number of samples that you have. So if you have 100 samples, maximum will return 100 features. If you do traditional rich regression, you will shrink, but you will not put things to zero, what Lasso will do, for example. Yes.
Right.
But there is this bias variance trade-off.
Right.
So you say, I do not mind to have actually a small bias if it gives me a better prediction because my variance is better approximated.
Right.
The issue is you do not know upfront how many features you need.
So in your work, you work with, do you have a problem where you have, what does a typical data set look like? A
difficult data set. Very vague description of what people want to predict, or very difficult to predict, yes, and not necessarily a lot of signal in the data.
Okay. So people just have a data set and they just sort of want to figure out if there's a pattern in there at all.
Yeah, like one of the things I've been doing in the past is, for instance, people who study dermatitis. Mm-hmm. You have different reasons for dermatitis. You can have a contact dermatitis, yes, because if you got into contact with a specific substance that you don't react well. Think about nickel, these type of things, yes, or copper. And you have a different version as well. Now, you have they called ICD, ACD, and they have different mixing forms, yes. The problem is these mixing forms to discern them. So they people that I worked with, they collected a lot of extra biomarker information, what they thought that could be difficult, yes, or interesting, yes. Mm-hmm. What seems to be the case? ACD versus ICD, so allergenic versus irritative dermatitis, yes, it's relatively simple to pull apart.
Okay.
However, the mix form is very difficult. Very difficult. Because you have something which in between, you have different things going on. Coming up with a proper machine learner or a proper set of features even, yes, that's super difficult.
Right.
So there is not much in the data to find these very little nuances, and also it's very difficult because it depends also on the person who's collecting the data to say, or the dermatologist, it's the mixed form or it's the pure form, for example.
Yeah, a lot of label noise as well in there.
Yes. So the noisier your data is, the harder it is.
Right.
Of course, the more satisfying it is if you get very noisy data and you're still able to extract it. It's a good feeling, yes? I mean, that's why you do science, yes? Because you want to also have that eureka moment to some extent, yes? But the hardest thing is, basically trying to understand what people want to do, like the doctors, what the molecular biologists try to do, and to convert that into a mathematical derivation or a machine learner or inference, whatever you are working on, that that answers that question.
Nice.
That's actually what I would say what my job as a biostatistician or a medical statistician is, or the job I do here.
Nice. That sounds. I mean, it sounds like you get to work with a whole bunch of different problems, and
that's also what you see. As you said, I did a lot of things. I did space things. I did immunological aging. I did microbiomics. That's what I've been doing as support.
Man, I really want to talk to you about the space and the immune age thing, but we have a few minutes before we have to go. But
yeah, well, if you want to, we can do it. We can continue another time if you want.
We can continue another time, but just like a cliff notes. The immune age thing I found very interesting.
Well, it was not my idea.
Yeah,
I got it so I do consult here as well. Yeah, so I got a question from somebody who was interested in it, and she just wanted methodological advice.
So okay, in this situation, right? How deep do you go into the actual biological problem? Versus, like, how do you constrain? Not constrain, but how do you scope the problem such that you can be effective without getting lost in the weeds of all the things you don't really need to get lost in the weeds for?
So, I do quite often abstraction.
Okay.
In theory, like you said, all the data sets that you see, yes, they're data matrix. Yes. Okay. So then the question is, how can I do something with all these rows that I have, all my variables that I measure? How can I do something meaningful for that person? Yes. And that person generally has some type of knowledge, and based upon the discussion you have, you decide, maybe this way, maybe that way. And you take a look on also the values in the matrix itself. What are the properties of these values? Do I expect certain interactions? Do I expect correlation present in there? And you build it up. I build it up from there.
What I find very interesting is this sort of interplay where, let's say you have a matrix of data, something collected from, and mathematics is all built on assumptions and built on axioms, right?
Unfortunately, yes, or luckily, maybe, depending on the person.
Yes, and for example, if you have this data collected from, let's say, a cell or whatever the context, and then you apply a mathematical transformation or an axiom, you can inadvertently imply an interaction or a behavior in the cell that the biologist would be like, wait, what? No, this makes no sense. Like, you can't have it this way.
Actually, this is maybe a sidestep, but I found that during my training as a statistician, I went to see a talk at ASMS, by the way, by Keith Baggerly. And he told a very nice anecdote. And he said, it was the time that people were doing microarray studies. So he said, let's see how well the biologists actually understand their topic. So instead of giving them the significantly different genes with different transcription levels, different abundance, let's give them a random list.
Okay.
So they went to the meeting with the biologist, and the biologist could immediately make sense of it, because that gene is in this pathway, that gene is in that pathway. So they were very happy with the results until they told them, we gave you actually a random set of genes, yes? We didn't do any test. We just wanted to see. This is not to make fun of people who do biology or molecular, but this is telling how complex the data also is.
Right.
So if you choose the wrong, and maybe not based upon the right foundations to do your model, you could still come up with reasonable outcomes.
Okay.
But why do we work with axioms and theorems and all of these type of things and assumptions? Because we believe that things work in a certain way. Based upon this, we try to draw these type of conclusions.
Right.
That's what we try to do, yes, as a statistician.
Right. So this is a super interesting sort of area for me, because I'm really thinking about the real world consequences of the mathematical decisions you make. For example, I did a project some time back where we were trying to predict the concentration of a metabolites in the cell over time, which is using a neural network and differential equations. So using a neural network to play the part of the derivative function. So you solve it with a
it would solve your complex differential equation.
Exactly.
Yes.
And I found, and then my supervisor at the time was working with adding physiological constraints to the loss function.
Yeah.
So what I did was, I penalize you for predicting negative concentrations, because that makes no sense. You can't.
No.
There's no negative concentration. So you don't do that.
That's one way to do it. What you do as a statistician, because you know it cannot have a negative concentration, you impose a certain transformation on that actually accounts for that.
Yeah, I find that super cool. I find that super interesting that you can just like that you take this reality of the world and then you think of ways to encode that into a mathematical model and a function to see if that holds or see if it helps with prediction.
Yeah, and that's what people very often get lost to. Just think about a normal distribution is continuous, and in theory it could go from minus infinity to plus infinity,
right.
yes? But very often biological data doesn't do that, yes?
Right.
And like, transcriptomics, you have count data. If you do sequencing, yes? People sometimes try to still use the normal distribution on top of that, but it's wrong. I mean, it's not necessarily wrong. There might be reasons why you do with a certain transformation, but generally it's discrete, meaning you have 0, 1, 2, but you don't have 1.2, 1.3, and so on anymore. That has completely different properties. It does not mean that if you do the normal, you get a wrong output. Your average will be quite okay, but it's the uncertainty around it that's not handled properly. And by that not properly handling, you are able to make mistakes, false positives, false negatives. That's why the choice of a right distribution is very important. But that's what's very often forgotten by biologists and by many people who look at the data and don't have that statistics training. You estimate two things. You estimate what you're interested in, but also how certain you are in it. So this is the precision accuracy type of thing, yes? Classification, it's not necessarily that important. But even then you try to say, how certain am I about that sensitivity and specificity? Which is also derived from the properties of your data, the number of samples, and all of these type of things as well, yes?
Yes. Nice. All right, let's pause here for now.
Yeah, okay, I know I digressed a lot about the mass spec stuff.
No, it's okay. I only got past like the first of questions because we went on so many tangents, but we will continue again for sure.
Yeah, if you want to, yes.
Yeah, of course.
Happy
super interesting.
We can also do it without the podcast.
Yes, of course. All right, man. I'll talk to you next time.
Yeah, okay. Thank you, Udesh.